All right, welcome Biochem students. This is the beginning of the Chapter 6 slides, and I just want to make clear we are going to work with the program PyMol on Wednesday. The setup.exe that you download from Moodle should be everything you need to just install the program. You don't need to go to the website where you log in and try to use the password that's provided on the read first instructions. If you have issues, then do that. But you should be able to just download the setup, click next, 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 finish, and then go to the protein data bank and download this 1FBY file, and the instructions for that are on Moodle, okay? Chapters 5, 6, and 7, as I already said, are kind of go together about protein structure and function and looking at the individual amino acids, which we already did in Chapter 5. These are the sections in the book that you should be looking at and that you should be reading so that you can understand this material. Okay, so this is just an outline that shows you the order that we're going to go through it. If you notice, Chapter 6 is kind of split up a little bit. So just to keep that in mind, there's the outline. Chapter 6 looks at 3D structure of proteins, and we're going to look at 4 levels of protein structure: primary, secondary, tertiary, and quaternary. So these 4 levels, you need to know the definitions, you need to be able to draw them, you need to know how to explain them, what's the difference, all of that. We're going to look at bond angles and primary structure first, and then we're going to go up from there. We're going to look at globular and fibrous probably on Friday or next week. Okay, so we'll do most of it today. All right, the 4 levels are primary, secondary, tertiary, and quaternary, and they're often abbreviated with the number and then the little degree symbol. So you need to know how to spell it. Oftentimes students misspell quaternary, but if you want to just, when you're practicing in your notes, just use the 1, 2, 3, and 4 with the degree symbol. That'll make it a lot faster and easier. So here are the definitions of the 4 levels. I'm going to go through them quickly, but you can always pause it to fill in the blanks on your slides. So primary structure is simply the amino acid sequence. That would be the single letter abbreviations or the 3-letter abbreviations just listed out. We don't know anything based on the primary structure about the 3-dimensional shape of this protein or anything like that, but we can just know what amino acids are present in the protein. The secondary structure is where we start to get 3-dimensional arrangement of these polypeptides. And there are 2 secondary structural elements that we're going to look at and what governs their 3-dimensional structure and that type of thing. Those will combine to form the tertiary structure, and that will actually be the first fully functional protein is tertiary structure. So multiple secondary structural elements form a tertiary structure that is a fully functional protein. Now, some proteins need more than one separate tertiary structure to actually function, and that is going to be the quaternary level. So more than one chain is what they're sometimes called, more than one domain is what they're sometimes called. That's quaternary structure, and we'll see examples of that at the end. Okay, so we already went over the peptide bond in Chapter 1, but this is showing it to you again. You should practice drawing this over and over and over and over again. You should know the peptide bond and just peptide amino acids just like the back of your hand. Okay, so as I said before, it's the amino acid sequence. The formation of this takes place in the ribosome, and the peptide bond can actually be broken by proteases or another class of enzymes called peptidases, and we're going to look at the protease mechanism in Chapter 8 later in the semester, but it can be broken. I just want to make that known. So the primary structure by convention goes N-terminus to C-terminus. N-terminus is the end. That's the amino group for, in this case, it's residue 1. So you might have 300 amino acids. The N-terminus is always residue 1. The C-terminus is harder because it could be, in this example, it's residue 4, but it could also be, residue 568 if the protein has 568 amino acids. So it's the carboxylic acid functional group. Just like our alphabet goes A to Z, proteins are read N to C, and since we don't look at the 3-dimensional structure in primary structure, since we're just looking at a series of amino acids, we need to know where the beginning is and we need to know where the end is. So you should practice drawing this. You should just pick any 4 or 5-letter word, single letter abbreviations, and just practice drawing it, not R1, R2, R3. Just actually practice putting this together because you're going to need to be able to do that on the next exam. Okay, so you may see a primary sequence with the 3-letter abbreviations, and that's just like this: glucir, phenylalanine, glycine, aspartic acid. Or it could be the single letter abbreviations like your book shows, where this segment that's actually in turquoise, they're going to make it into a secondary structural element, so you can see the difference. So primary sequence just looks like the N terminus to C terminus, sort of beginning to end. No three-dimensional world shown in this image. Okay, I want to focus on the peptide bond for a second. So this is the peptide bond in the sort of upper left-ish area. It actually has resonance. The lone pair on the nitrogen can move its, the lone pair on the nitrogen can move into the bond between the nitrogen and then the carbonyl carbon, and that actually forms this structure on the far, far right. So remember, these double arrows from organic chemistry, that's a resonance arrow. So this image on the right is a perfectly legitimate way of drawing the peptide bond. It's a harder way to draw it, but it is accurate. It also has a partial charge. If you notice, there's a positive on the nitrogen and a negative on the oxygen. So the peptide bond has partial double bond characteristics due to this resonance. You need to know that. And because of this, you actually cannot rotate around the peptide bond. It's impossible to rotate around that partial double bond. It's a lot more rigid than a single bond. So that limits the number of conformational possibilities that we're going to see in the 3D world for the three-dimensional structure of proteins. This shows the peptide bonds in red, and then they're kind of, if you're looking at all of the atoms that are participating in the peptide bond, they're all on the same plane. So they've tried to show in this image, they've sort of boxed in green 3 peptide bonds. Now the N to C alpha bond, that is known as the phi bond, and that is rotatable. You can rotate around that bond. The C alpha to the next carbonyl carbon is known as the psi bond, and that is also flexible. So you can rotate around the phi or the psi bonds, and those angles will actually form the three-dimensional structure of proteins when we look at secondary structural elements. Okay, so you can rotate around those. It is possible. You just cannot around the peptide bond because of that double bond from the resonance. Okay, so up here at the top is the primary structure, and what your book shows is just an image where that series of turquoise amino acids is now a three-dimensional secondary structural element. And the formation of this is the rotation around those phi and psi bonds, and it's limited by steric interference. So larger side chains are going to clash with each other, so they're not going to be in the same space. It's most importantly governed by hydrogen bonding, and it's hydrogen bonding often in the backbone, and there's also some interaction with the side chains, which we'll see. So there are 2 secondary structural elements, the alpha helix, you need to know that term, and the beta sheet, you need to know that term. And the alpha helix usually kind of looks like the helical corkscrew, and then the beta sheet is represented by arrows, usually sort of flat arrows, and these are connected by loops, which I kind of colored it gray, and it looks just like limp spaghetti. So loops are just connecting the 2 main secondary structural elements. So we're going to focus on the alpha helix first. Okay, so the carbonyl carbon forms a hydrogen bond with the amine hydrogen. Okay, and that's going to be 4 amino acids later. The helix is right-handed, and there are approximately 3.6 residues per turn. And the side chains actually extend outward. So if you were looking sort of top-down, if it looked like the circle, side chains would be pointing out like a child would draw the sun. Okay, so if you actually look at this sort of more purple image, here's an example of a hydrogen bond between carbonyl oxygen, hydrogen bonding with an amine hydrogen on the backbone, 3.6 residues later. Okay, so the image on the bottom is just showing sideways. The image on the right is just showing N to C terminus with hydrogen bonds as sort of the yellow dashes, and the hydrogens aren't shown on the amine, so you can see the yellow dotted line is just looks like it's going straight to the nitrogen, but the hydrogen is just not shown in the image on the right. Okay, this is a diagram that I made. It's not from your book, and you start in the upper right and you sort of read this way, because remember it's right-handed. So if you're curving to the right, we go from N terminus, so carbon or alpha carbon for residue 1, 2, 3, and then you are into the board, come back around, and you're over here back out into the front of you, 3.6 residues later. So this, if this shows the hydrogen bonding better, kind of going up and down, it's all in the backbone, hydrogen bonding, okay? And the side chains would be coming out. So here, if you look at the image on the right, the side chains, can they interact with each other? Yes, but you need to know who can interact with who. So if you're looking down, residue 1 here is actually close to amino acid 5. It's also available, depending on how large they are, to interact with residue 4. So 1 could have maybe a charge-charge attraction with residue 5 or residue 4, or it could have van der Waals attractions, those other attractions that we talked about earlier in the semester. They can interact with each other, but 1 and 3, for example, can't interact with each other. Another comment on the alpha helix is that proline is too rigid to participate in an alpha helix, so it's often not seen in an alpha helix. It's called a helix breaker. And then glycine, because it's way too flexible, because it's just a hydrogen side chain, is also often not seen in an alpha helix because it's too flexible. So it's also known as a helix breaker. Okay? So that was the alpha helix. The beta sheet is represented by an arrow. So we have the beta sheet that is more than one arrow, and then we have the beta strand. Okay, so a single arrow would represent a single beta strand. You need to know the difference between strand versus sheet. Sheet is going to have more than one arrow. Oftentimes, there's more than one arrow. But in a single beta strand, you could talk about, oh, this one amino acid located in this beta strand. You wouldn't necessarily have to say beta sheet, okay? So, there are two types of beta sheets. There's parallel, where the arrows are pointing the same direction, and anti-parallel, where they're pointing opposite directions. If you're trying to figure out where the N terminus and the C terminus is, the arrow always points toward the C terminus. Okay, so I sort of drew it there on the arrow that was by itself. And it forms a pleated shape in both, which is easy to see in the images in the coming slides, but in both cases, the side chains are going to extend up and down. So based on the arrows that are here, the side chains would be coming out at us, or into the board away from us. Okay, so there on the top is an image that shows the arrows in green of an anti-parallel pleated sheet. So you can see the pleats that are kind of pale purple and dark purple. You can also see the R groups pointing out. So this is above the arrows, or down, sort of hidden away, down. And based on the chirality of carbon, you can start to see why it's pleated, because carbon often has sp3 hybridization. This is an alpha carbon and it has sp3 hybridization, so it's going to be pointing up, and then the next alpha carbon is pointing down. The hydrogen bonds go between the arrows on the sheet, and they're the same concept that we saw with the alpha helix. It's hydrogen bonding in the backbone. It's the amine hydrogen with the oxygen on the carbonyl. So there's hydrogen bonding. The image on the top did not come from your book, and the image on the bottom did. And the hydrogen bonding on the bottom is shown as the yellow dashes. Okay? I made my own image of a anti-parallel beta sheet showing the hydrogen bonding. If you look at the hydrogen bonding here, it kind of looks like two next to each other, a little gap, and then two more next to each other here. Okay? So you can see the different types of hydrogen bonding here with the anti-parallel beta sheet, sort of in detail, zoomed in. And then if you actually compare anti-parallel on the left, it looks pretty simple. But on the right, parallel, we don't line up quite so pretty. The hydrogen bonds are at an angle. They still exist, but they're slightly angled. You should be able to look at a beta sheet and know whether it's parallel or anti-parallel based on the hydrogen bonds. Okay? So we got three images here showing the different hydrogen bonds, shows the N to C terminus. You can look at these slides, the images, they came straight from your book. One little extra thing you may read about when you're doing your take-home quiz is that the secondary structure, sometimes if you have a special loop on a beta sheet, it's called a beta turn. Sometimes people just call it a loop. Okay, it's 4 residues long, and 1 and 4 may interact with each other in a hydrogen bond. So that's the carbonyl oxygen of residue 1, once again with the amine of residue 4. It's a beta turn, it's flexible, It's not part of the sheet, but because it connects two beta strands, it gets a special name.